In the last module, we talked about what stress is on a molecular level, and we started talking about the free radical theory of aging. Let's look into this a little bit more in detail in this module. In cells, sugar is oxidized. In other words, it's burnt, in effect, in order to create energy. The flame that can be seen when you burn sugar is indicative of the energy that is present in sugar. In the body, this is achieved without waste, that is, without loss of energy as heat or a flame, in the normal biochemical reactions that make up cellular energy metabolism. Cellular metabolism of most eukaryotic cells is critically dependent on the presence of oxygen. Without it, the majority of energy that is in a sugar molecule could not be utilized and converted into cellular energy, or ATP. But the same side effects that uncontrolled sugar burnings can have, for example, you set your hair on fire or your clothes or your apartment, can also occur within a cell with equally disastrous consequences. The realization that oxygen, while absolutely required for the majority of advanced life on Earth, is also at the same time one of the most toxic and harmful molecules, led to the formulation of the free radical theory of aging. As we've mentioned before, the majority of cellular energy is created in the mitochondria through a process called oxidative phosphorylation. In this process, electrons are passed on between the enzymes of the electron transport chain in the inner mitochondrial membrane, which ultimately leads to the creation of ATP. The final molecule to take up these electrons is molecular oxygen, which is turned into harmless water in the process. However, these reactions are not perfect and sometimes create ROS, such as the superoxide anion, hydroxy radicals, or hydrogen peroxide H2O2. If you have ever used hydrogen peroxide, you know how damaging this molecule can be. In chemistry, a radical is defined as a molecule with an unpaired electron that is incredibly reactive. Thus, creation of ROS by mitochondrial metabolism creates molecules that can attack, modify, and quite possibly destroy cellular macromolecules, from lipids to proteins and even nucleic acids. What is shown in this example is lipid peroxidation, the destruction of lipids. Reactive oxygen species, oxygen radicals, can attack lipids, especially at electron-dense structures such as double bonds. That creates lipid radicals, which in turn can attack other lipids, thus leading to a chain reaction that results in the destruction of these lipids. These lipids are broken down into smaller molecules and eventually are completely destroyed. In order to counteract this cellular damage, cells have evolved very sophisticated defense systems. We talked about those before, such as the glutathione system, the SOD, and the catalase system that detoxify those reactive oxygen species into harmless water. Many substances outside the body, so-called xenobiotics, may also lead to the generation of radicals, compounding the effects of metabolic ROS generation. This cellular damage, caused simply by the very act of living, provides a very elegant explanation for the aging process and fits neatly as a mechanistic explanation into the evolutionary theories of aging. Aging simply occurs because damage caused by ROS over time cannot be repaired, leading to functional decline of molecules, therefore cellular macromolecules, and therefore in turn organismal structures. And that is what aging is. That was one of our definitions of aging. Organisms have only limited resources for repair processes. So these resources could be shifted to the maintenance of germ cells, which is what, for example, the disposable soma theory states after all. Not only does this make intuitive sense, some experimental data also is in support of it, as there seems to be a clear inverse correlation between mitochondrial ROS production and longevity. For example, if we compare different types of animals with their mitochondrial ROS production, at least for vertebrates, the amount of ROS produced by mitochondrial is negatively correlated with maximum lifespan. Similarly, reducing dietary food intake, a treatment that is known as calorie restriction or sometimes called dietary restriction, is very clearly associated with reduced cellular damage, and that makes sense. In this table, we see the kind of treatment of dietary restriction that particular animals are undergoing. And in all cases, oxidative damage, either in the form of lipid damage, protein damage, or even mitochondrial DNA damage, is reduced in animals that were exposed to this dietary restriction regime. So here we have a beautiful, clean, and elegant theory that is not only able to explain why, but also how aging occurs, and even better yet, also is in accordance with many experimental results. So appealing is this idea that many non-scientists have even heard of it and have bought into it wholesale. One needs to look no further than the supplements aisle in your local supermarket to see the plethora of antioxidants that is being sold there, from vitamin C and E to green teas, catechins, and substances with even more exotic names. Since this theory, well, let's call it properly a hypothesis, has been around since the 1950s, one would think that within 60 years, someone would have developed a longevity pill based on it. That this is not the case reveals one important flaw of the FRTA, that it is most likely wrong. At least in its current form. While the data on ROS production and lifespan implies causality, it's merely correlative. It is notoriously difficult to properly measure ROS produced from mitochondria. It is usually done by extracting the mitochondria from living cells, followed by measurements using artificial substrates in a test tube. Depending on the substrate, the isolating method, the precise measurement conditions, experimental results may be skewed and not accurately reflect what is actually going on inside the cell. And directly measuring ROS production in vivo, in live organisms, is even more difficult. Thus, the real test of the theory is a direct manipulation of ROS levels or ROS detoxification systems and a subsequent evaluation of the resulting lifespans. Over the last decades, many such tests have been performed, and they all have failed to show causality. One such approach is to artificially increase antioxidants defenses by overactivating the enzymes involved in detoxification. In C. elegans, no positive effects on longevity can be demonstrated when any of the detox enzymes are overexpressed. In Drosophila, positive effects are only observed in very short-lived strains or depend on a particular experimental approach. In mice, overexpression of SOD, the key enzyme in detoxification, does not extend lifespan, while catalase may extend longevity when targeted only to mitochondria. Similarly, antioxidant supplementation should have positive effects on longevity, but the data are inconclusive at best. No antioxidant tested to date, whether that be vitamin C, vitamin E, green tea, antioxidants, and so on, has reproducibly and consistently extended longevity in model animals. On the other hand, reducing antioxidant defenses by disabling genes such as SOD, catalase, GST, etc., fairly consistently shortens animal longevity, as does increasing atmospheric O2 concentrations, an intervention called hyperoxia. So, clearly, ROS can influence disease and longevity, and are often key factors in diseases, especially neurodegenerative diseases, diabetes, or even cancer. But they are not the only factor in aging, and anti-ROS therapy, therefore, is not a panacea to treat aging. Does that mean the free radical theory of aging is dead? Not necessarily, but modifications are certainly in order. One such modification stems from the observation that ROS can be deliberately produced by cells to act as signaling molecules, small molecules that act in cell-cell communication and elicit changes in gene transcription and cell function. One such modification of the FRTA is called hormesis. Hormesis is the idea that small amounts of stress are actually beneficial to an organism by inducing a much more robust defense response. Thus, at low concentrations, ROS would function as signaling molecules, alerting a cell to the presence of potentially dangerous environmental conditions and bolstering cellular defenses. This concept is similar to how the immune system functions, where it needs exposure to pathogens to become fully functional. This is the very same concept vaccinations are based on. If hormesis is true, then small amounts of stress should lead to increased lifespan. In this experiment, C. elegans were exposed for a short amount of time to high temperatures. Their normal rearing temperature is around 15 to 20 degrees Celsius. In this experiment, they were pre-treated up to 35 degrees Celsius for several hours. As predicted, short pre-treatment at elevated temperatures did in fact extend warm longevity, while longer treatments increased stress levels too much and shortened lifespans. Other hormetic stimuli include cold, electric shock, or radiation. Hormesis thus appears to be a generally applicable concept when it comes to stress. Importantly, hormesis may explain why antioxidant supplementation often show detrimental effects in model animals. Antioxidants may suppress the hormetic response and paradoxically lead to lowered defenses and shortened lifespans. The molecular basis for the hormetic response is still quite unclear and currently an intense focus in molecular aging research. One of the best understood aging pathway is the IAS that we talked about earlier. The details of the IAS vary by species, but it consists of a cell surface receptor for insulin-like proteins, intracellular effector molecules such as PI3 kinase and protein kinase B or AKT, and DNA binding proteins of the FOXO class that act as transcription factors and can modulate gene activity. In C. elegans, these molecules are called DAF2, that's the cell surface receptor, H1, that's a adapter molecule called a PI3 kinase, AKT, and DAF16. Inhibition of this pathway extends longevity in many model species, but we will talk about this in more detail when we talk about the endocrine system. Interestingly, DAF16 controls, amongst many other genes, the transcription of small heat shock proteins, such as the small heat shock protein SHSP16, that function in the anti-stress response. As a side note, the name DAF is derived from the process of Dower formation, which is a C. elegans developmental process that creates the so-called Dower stage, an extremely long-lived, developmentally arrested, stress-resistant larval stage that is induced in response to poor environmental conditions. Many genes involved in this Dower formation pathway have been shown to increase stress resistance and longevity. Another variation on the theme of hormesis is the so-called mitohormesis, or hormetic response specifically involving mitochondria. Mitohormesis was first observed in cells that were glucose restricted, that is, had less glucose available for energy metabolism. Instead of showing reduced ROS production due to those lower glucose levels and therefore slower metabolism, they showed an increase in ROS production, suggesting more efficient mitochondrial metabolism. This in turn had hormetic effects on stress response and longevity. Many known aging signaling pathways or aging interventions, such as dietary restriction, TOR signaling, a pathway that is important for protein synthesis and protein quality control, may elicit hormetic responses. In addition to ROS production, mitohormesis also involves the mitochondrial unfolded protein response, a pathway that specifically deals with unfolded and denatured mitochondrial proteins in order to restore those back to the correctly folded and fully functional form. It is possible that many longevity pathways may act through mitochondria, and we will talk about mitochondria specifically a bit later in this class. While ROS in and by themselves may not be causally involved in the aging process and may constitute only one of several factors that contribute to it, they're clearly associated with disease states and even to a degree with the aging phenotype itself. In small doses, ROS elicit a hormetic response and or a mitohormetic response that boosts cellular antioxidant defenses and may contribute to lower aging rates. At higher concentrations, ROS damage cellular macromolecules and contribute to the development of disease states, which is not the same as accelerated aging. Let's be very clear about it. Animals that are short-lived due to increased ROS damage may simply be sick. But stress and ROS levels are higher in old animals, which could be used as a predictor for remaining lifespan. In other words, ROS could be biomarkers of aging. In the first attempt at such a diagnostic tool, the age-dependent levels of the stress responder SHSP 16.2, the small heat shock protein that we were just talking about, were measured in C. elegans using a simple microscopic assay. This protein is almost non-detectable in young animals, but levels increase as the animal gets older. However, gene expression and longevity is a stochastic process, meaning highly individualized to the point that even identical twins have different lifespans. The basis of the stochasticity of the aging process is still unclear. But what the C. elegans experiments revealed is that animals at the high end of SHSP expression had longer lifespans of 15 days or more, shown in the green bars, while animals at the low end of SHSP expression had very short lifespans, 5 days or lower, as shown by the red bars. Average expression led to average lifespans. These data highlight two things: that stress response systems are clearly important for aging phenotypes, and that it may be possible to use these systems for diagnostic and even predictive purposes. Quite a bit of research is going on that front, but it remains to be seen if it truly can be translated to humans. Check your understanding. Describe the evidence for and against the FRTA. Define hormesis and mitohormesis.